Hello everyone, I'm Yifeng, currently a PhD student from Carnegie Mellon University. It is my pleasure to introduce our work, Robust and Accurate Deconvolution of Tumor Populations Uncovers Evolutionary Mechanisms of Breast Cancer Metastasis. This is the joint work with Haoyun Lei, Xuechang Fu, Dr. Adrian Li, Dr. Jian Ma, and Dr. Russell Schwartz. Tumor does not consist of a single cell type. During its progression, tumor cells follow a clonal evolution process leading to heterogeneous populations even from the same site. At some point, some cancer cells may leave from the original primary site and move to other sites of the body, which is a phenomenon called metastasis. Breast cancer is the second common cause of death from cancer in women. Among all the cases of breast cancer, metastasis causes the majority of these deaths. The metastatic sites can be brain, gastrointestinal, ovary, or bone for breast cancer. Once metastasis happens, there are usually only limited viable treatment options. Therefore, the early detection of breast cancer metastasis is critical. Understanding the mechanism of tumor progression during metastasis relies on phylogenetic analysis. Single-cell sequencing seems to be a plausible tool for this goal. However, they are usually not available due to yields between sample collection. So, how to robustly and accurately unmix the bulk tumor samples is essential for understanding the tumor evolution of breast cancer metastasis. In this work, we propose a toolkit called Robust and Accurate Deconvolution, or RAD, to address the issues in unmixing bulk RNA data of primary and multiple metastatic tumors from a panel of cancer samples. In this work, we used the bulk transcriptome data of both primary and metastatic sites from cancer patients. In order to fully utilize the power of RAD, we first compress the genes into gene module through knowledge base, which in our case is DAVID. The genes in the same gene module are more likely to co-express. This step effectively reduces the noise in the data. Secondly, we apply the core algorithm of RAD to deconvolve the bulk sample. And finally, based on the RAD on mixed tumor cell populations, we infer the evolutionary trajectory through MEP algorithm. We formulate the RAD as a non-negative matrix factorization problem. The input of the algorithm is a bulk transcriptome of both breast primary and metastatic samples, represented as the matrix B. The output of RAD are two non-negative matrices C and F. C represents the RNA expressions of each cell population, and F represents the fractions of each population. We have an additional normalization constraint of F compared with standard NMF problem. This constraint is essential to make the RAD model biologically meaningful, because we want the fractions of all cell types in a single sample always sum up to 1. The problem itself is hard in a few aspects. First, the RNA data is usually noisy and correlated. Therefore, the solver needs to be robust to the noise. In this work, we introduce gene module compression to alleviate this, which will be shown later. Second, the problem itself is non-convex, and as far as we know, there does not exist an efficient optimizer for this specific problem. Lastly, the number of cell populations, or K, in this problem is provided as a hyperparameter. However, we usually do not know this number in advance. The core algorithm or RAD consists of three phases. The first phase is warm start, which follows the revised multiplicative update rules. It iterates the MU rules to update matrices C and F until the objective function stops decreasing. It is the revised version in the sense that we have an additional normalization step of matrix F. Notice that MU rules can only guarantee the objective function is non-increasing in the general NMF problem. The objective may increase in our case. However, it is still a super fast approach to converge to a reasonable solution in our case. The second phase is coordinate descent. We optimize the C and F by fixing the other one iteratively until convergence. Although the original problem is non-convex, the subproblems of coordinate descent are convex and can be solved as quadratic programming problems. The coordinate descent is more computationally expensive compared with a multiplicative update warm start. However, this phase further reduces the objective loss by around 5 to 30%. The last phase of RAD is minimum similarity selection. It aims to reduce the randomness of different initialization and select the most reasonable solution from different runs. In order to realize this, we repeat the random initialization, phase 1 and phase 2 for multiple times, for example 10 times in our default setting. Then we select the solution with minimum cosine similarity among all the 10 solutions. According to our criteria, a good solution is where cell components or populations are roughly orthogonal from each other. As we mentioned before, the number of cell populations is a hyperparameter at the time of optimization, which is, however, usually unknown to us in prior. We propose to utilize cross-validation to select the K that achieves the minimum cross-validation errors. The trick we take is masking. For example, if we take 10-fold cross-validation, we create a binary mask matrix M at the training time. 90% of the elements are set to be ones, but others to be zeros. Therefore, we only optimize according to the 90% of the elements of the original bulk data. At the validation time, we use a complementary mask for calculating validation error. Although we do not show here, there exists a masked version of RAD optimization algorithms. To evaluate the effect of gene module compression, the RAD accuracy on estimating C, F, and K, and to understand the breast cancer metastasis mechanism, we explore three distinct datasets in this work. The first one is simulated dataset, where the gene module and ground truth C and F are known to us. We can generate these datasets repeatedly to draw a reliable statistical conclusion. The second one is GSE dataset. It is a mixed RNA expression of rat brain, liver, and lung samples. The ground truth C and F are known, but we have to infer the gene module through knowledge base, for example through the tool David in our work. The last one is the breast cancer metastasis dataset, which consists of bulk RNA data of both matched primary breast and metastatic samples. We do not know the ground truth C and F and we have to infer the gene module through knowledge base as well. Here, we first explore the effect of gene module size on the deconvolution accuracy through the simulated datasets. We use correlation of estimated and ground truth values as accuracy metric for both C and F. When module size equals to 1, it is equivalent to the original gene representation without any module compression. We can see that a deconvolution using the original gene representation or a too small module size is prone to fragile deconvolution. On the other hand, if the module size is too large, it can also result in worse estimation. Proper module size is helpful in improving the model performance. Secondly, we found RAD can detect the correct number of cell populations. Here we plot the curves of cross-validation errors in both the GSE and BIM datasets. We can find that the cross-validation error in GSE drops quickly before k equals to 3 and almost stops decreasing after that. Actually, we do have 3 cell types in total in the GSE data. For BIM datasets, the cross-validation error reaches its minimum when the number of populations equals to 7 and increases further after that. We therefore choose 7 as the default hyperparameter for BIM data. Here we compare the RAD method with a few other competing algorithms, including geometric unmixing, linseed, and NND. Both aim to resolve the complete deconvolution problem. We use the correlation as well as L1 loss and MSE as accuracy metrics of estimation. We can find that the RAD algorithm, together with gene module compression, is able to outperform the other 3 methods on GSE data. We have shown in the previous slide that proper module size can improve the RAD performance in simulated datasets. Here we also implement the RAD version with the raw gene expression instead of gene module. By comparing column D and column E, we can find the gene module is also helpful in the GSE data, even when the modules are inferred from knowledge base. Finally, we infer the phylogenies from the mixed cell components. We use the minimum elastic potential method, which is discussed in our previous work. Since not every cell population exists in all the cancer patients, we have 4 cases in total. Here we only show one of them. However, there exist common early pathway level events from these 4 cases. That is the decrease of PI3K-AKT pathway, the decrease of ECM receptor interaction, and the decrease of focal adhesion. Previous research have shown the loss of PI3K-AKT pathway in metastatic tumor through both genomic and transcriptomic data, which is not a surprising result here. Tumor cells adhere to the extracellular matrix, forming the structures called focal adhesions. Our result here indicate the loss of these interactions is a key step in enabling metastatic migration. In conclusion, deconvolution of bulk data is the key to understanding the breast cancer metastasis progression. In this work, we propose RAD, a toolkit that accurately and robustly estimates the number of cell populations, expression profiles of cell populations, and fractions of the populations. Through RAD, we find the loss of PI3K-AKT ECM receptor interaction and focal adhesion emerge as the common early pathway level events of breast cancer metastasis. Although it is hard to collect single-cell data from primary samples, it is possible to access them for metastatic ones. Therefore, it will be interesting to explore how to integrate this partial single-cell data to improve the RAD performance in the future. I would like to thank my advisor, Dr. Russell Schwartz, our collaborators, Dr. Jian Ma, Dr. Adrian Li, and my colleagues Haoying Lei and Shechong Fu. Without their help and devotion, the introduced work would not be possible. Haoying and Shechong will also have presentation at ISMB on tumor evolution. Feel free to check and join if you are interested.